Hi, welcome to the Carney Lab. A lot of our research is focused on extracellular vesicles, which are small nanoscale packages that are shed by every cell in the body. We use these to analyze a lot of different disease states, primarily cancer. In our lab, we have a lot of different instruments with biophotonics and analytical means that we can assess these EVs. So to start with, we have these two really big -80 freezers, and in these we keep our clinical samples where we isolate the EVs from. The first thing that we want to do when we get these EVs is do some general characterization of them. So this is our NanoView ExoView, which we can use to tell us how many EVs we have in the sample that we isolate, as well as the different tetraspanins, which are these proteins that are found on the surface. Next, we have our NCS1, which is a spectrodyne. This is a relatively new instrument that we have, and this uses resistive pulse sensing to tell us the size as it passes through an electrical current. Our main analytical tool in our lab is our Raman microscope. With Raman, we can collect inelastically scattered photons as they interact with the EV samples. And this can provide global chemical analysis about the different proteins, lipids, carbohydrates, nucleic acids, what have you, that are present on the EV surfaces. One of our goals for this is to see if we can distinguish between cancerous and non-cancerous EVs to potentially use it as a liquid biopsy diagnostic tool. Is pretty quirky, so one of the things that we're very proud of is calling our laser and our Raman system the Death Star, which we have a 3D printed one here. So over here we just have some bench spaces that our students use. So right here, this is where our student Marissa makes liposomes. In the back of the lab we have a couple tabletop centrifuges that we use for the isolation of our EVs. Back here we have some microfabrication setup, very fancy scientific milliQ system, which actually has been featured in Star Trek. That's one of Randy's favorite facts about our lab. Marissa here is using our Raman scope software and everything on that computer. So another thing that we do in this lab is nanoparticle synthesis, which Hanna, the other Hanna who's filming this actually, does a lot of. We use this hood to make these nanoparticles. We can make them in a lot of different shapes, and they're really good for the Raman signal. We have our new rotovape that we have set up. Again, they will be used for nanoparticle synthesis in the future. So this is just our run-of-the-mill scale. And then additionally we have our size exclusion chromatography setup, which is also used as an isolation technique for our EVs. And then the last part of our lab is this little side room right here. In here we have an instrument called the NTA, or nanoparticle tracking analysis. With this we can send our EV samples through, and it can give us a concentration and size distribution. So this is kind of a good step to know how many particles we have to start, and that's really important for a lot of therapeutic and dosing applications. And then we have a couple more big centrifuges on the tabletop here, and then our big guy right here, which again is just used for our EV isolation. That's an overview of our lab. I hope you guys enjoyed it. We are always open to taking more questions, and hopefully we'll be able to show it to you in person one day.